All right. Well, how's everybody doing? Welcome to my hometown of Philadelphia. And I just wanted to bring up Debbie Morissette. Debbie wrote that song and sang it. That's her voice you heard. How great was that?
How great is that?
Huh?
How great is that? And does them all. Does them all.
She sings almost all of them when you hear a female voice. So thank you.
Thank you. And I'll let you.
Anything you want to say about the inspiration here?
The inspiration was the one and only Roger McIntyre. If you noticed all the articles that were in there, those are all.
Those papers were real.
Yes.
Great job. That's wonderful.
Thank you, Deb.
Have fun with this.
We will, Deb. Thank
We
you.
will.
Good evening, everybody. Good evening and welcome.
Whoa. Oh, here we are
Andy,
again.
good to be in your hometown.
Good to be here. So, Roger, we're going to take a little walk down memory lane.
Yeah.
Now, Roger and I have something in common. We've both been very interested in the interaction of the brain and the body for many years. As a matter of fact, I did 2 residencies. I'm board certified in internal medicine and psychiatry because early on in my career, I was just absolutely fascinated. The same neurotransmitters in both places, all these interactions. But Roger, tell me, how did you first get interested in this?
Well, I'm sort of like Andy in the sense that I had started off in medicine and surgery before I actually did a deed school.
Hang on. You started in surgery?
Yeah, I did, actually. What had happened is I had, right from my very first year of medical training, I had, at that time, decided that I was going to go into neurosurgery.
Neurosurgery, that's what you told me.
And for the ensuing 4 years, I had orchestrated my entire training around that inevitability as I saw it, only then for the last rotation, literally, of medical school.
Yeah.
Was psychiatry, and that rotation was just a checkbox just to complete my training and move on to bigger and better things in life.
Yes.
Only then, in fact, to be self-aware, at least, that I was already becoming bored out of my mind in neurosurgery, doing what I was doing. And a patient was brought in by the law enforcement in my home province of Nova Scotia, Canada. And that gentleman had bipolar mania, and I had never seen that before clinically. And what's interesting and pertinent to today's conversation, Andy, that gentleman was morbidly obese. And when I was told that this person actually had bipolar 1 disorder, first time I'd ever seen that, one of my most favorite pieces of music is the Rachmaninoff 3.
Oh, yeah.
And I had almost in a sort of a synesthesia, so to speak. I started to hear the music, and I said to the resident, the chief resident, this person's got what looks like Rachmaninoff 3 to me. And the chief resident said, I don't know what the hell you're talking about. And I said, well, this is just unbelievable. And so I quit neurosurgery, and I went into bipolar disorder research. Here I am a few years later.
Yeah, that's an incredible story. And I share something with you too there. Psychiatry was the absolute last thing that I wanted to do when I went to med school. But funny thing happened, I just fell in love with the brain. It was just so damn interesting. Yeah.
Well, it was, and I think what happened when I first started off in psychiatry and training, I, of course, made an observation everyone in the room has made, and that being that our patients who are living with depression and bipolar disorder, not only are they more likely to be affected by obesity, type 2 diabetes mellitus, metabolic syndrome. We see this every day, obviously. That was not a unique observation. But what I noticed is that when I spoke to persons who had these comorbidities, when compared to those who didn't have those comorbidities, those that did were more likely, at least what appeared to me, which was later verified, was they more likely had cognitive problems. And in addition to the cognitive problems that they seem to have much more pronounced manifestations of, they also, in fact, seem to have a greater alteration of what you and I would call anhedonia, in the sense of more motivational deficit, reward deficits, and behavior suggestive of that. And it was always curious to me, and it kind of intellectually interrogated me, and it started getting me thinking that there may be a mind or brain-body connection, and it inspired me to sort of think about this as a framework where I kind of coined this phrase that obesity metastasizes to the brain. And I started to wonder whether or not there was, in fact, a crosstalk. And I always thought psychiatry is the profession of brain preservation. And anything that actually threatens brain preservation, I always thought would be a critical target for us in our enterprise of helping our patients. And I'd never actually had pre-thought that I'd be spending the next 30-odd years studying obesity, diabetes, and the brain.
You know what's interesting about that? We're now learning, interestingly, at a neurobiologic level that many, if not most, of our treatments are probably working through some degree of neuroprotection or neuroplasticity. So you were onto something there.
Well, you know, it's interesting. These things sometimes are a bit of luck. The way that, you know, as the hockey analogy goes, it's sort of where the puck is going.
Yeah.
I didn't know the puck was going there, and you know, people often say, "Who's your daddy?" I often say, "Who's your nana?" And nana is N-A-N-A, neuroplasticity, apoptosis, neuroprotection, and autophagy. What the hell is autophagy?
Oh, that's beautiful.
And that is that our cells have to self-repair and kind of eat away their bad hedges. And all these processes of NANA are all impaired, and that's the prevailing model. And what's so interesting is that I started my training in Toronto, Canada, 51st state, by the way. I don't need to get a passport anymore, so it's been great.
Yeah, it's good news.
The 51st state stuff has been great, because I was planning to get a passport to the U.S. anyways. Now I can just forget it. Anyways, on a serious note, in Toronto in 1921 was the first patient in the world who ever received insulin for the treatment of type 2 diabetes mellitus at my hospital. And so Toronto had a history of diabetes research, and many years later, in 1986, Dan Drucker and other investigators in New York and Copenhagen discovered the GLP-1 protein.
Really? That long ago?
Yeah. So it's a bit of a Toronto kind of, I guess, epicenter for diabetes research.
Yeah. So you are one of the first people who taught me that phrase, obesity metastasizes to the brain, but then you also taught me that diabetes metastasizes to the brain. So what started happening in your interrogation of diabetes, not just obesity?
You know, it's funny how it is. You know, like a lot of things in life, if you go into your closet, you start digging things out, you start realizing, wow, I didn't know this was here. I didn't know that was here. And so when we started looking at this in the brain, and trying to understand what were the mechanisms that could possibly explain the highly replicated observation that diabetics are more susceptible to cognitive impairment. They're more susceptible to MCI and major neurocognitive disorders. And just like the patients that we see, the cognitive impairments that the diabetic population have are multi-domain, executive function, memory, processing, speed, attention, but they're also progressive. So what was interesting was that the cognitive manifestations of diabetes are almost interchangeable with bipolar and major depression, and progressive. And then to add to that, what was also interesting as we started looking at this is that we all know that insulin's role in glucose homeostasis in the periphery, the liver, the skeletal muscle, so on and so forth. But in the human brain, the insulin protein and its receptor are not there for glucose utilization. The neurons in the glia can directly utilize glucose in the absence of insulin to enable its cellular entry. So then the question is, well, what's it hanging around for? Mother Nature is clearly not in the game of redundancy, and she's obviously an efficient engineer. So it turns out that when you knock down or knock out the insulin receptor, in other words, you make it just not functional or non-existent, what happens is that the neurodevelopment goes awry in the brain. And you also, in addition to seeing alterations in development, such as neurogenesis, the very pathophysiologic models that we implicate start to show up. You start to see alterations in neuroplasticity. You start to see premature cell death, which is apoptosis. You see altered brain repair called autophagy alterations. And then to add further to the storyline, when you knock out the insulin system, you start to see a massive deposition of amyloid and also tau protein, and another one called alpha-synuclein. And we all know that Parkinson's disease is a lesion in the dopamine, those tight, compact, pars compacta neurons in the substantia nigra. But the molecular hallmark of Parkinson's disease is alpha-synuclein deposits, which are aggregated more in diabetics. So we went one step further, and then we discovered by looking at the postmortem brain of people living with depression and bipolar disorder that there was something that was, there were these functional variants in their genes in the hippocampus prefrontal cortex for the insulin receptor.
Wow.
And then what we had discovered, we met a colleague at NIH who had validated this really interesting methodology where we can look at the brain's insulin sensitivity directly, because obviously patients are not going to give us a brain biopsy, right? A postmortem brain has its limitations from a methodologic and a translational perspective. But what we can do now is that we can take what's called exosomes, and these are neuronally derived vesicles that are in our blood. They can be separated from your blood, and they literally are derived from the neurons, and they are, in many ways, a microenvironment of your brain. And we discovered that on these vesicles, the insulin receptor was reduced in its sensitivity to insulin. So that was the first evidence of insulin impairment. Then, Andy, what we did was we moved to getting patients to start snorting insulin. We asked them to put up their nose, just snort it. And what we did by doing so in bipolar and depression was we were able to improve their cognition and reduce their anhedonic function. And this was subsequently followed by later work with metformin. Now, metformin's often called an insulin sensitizer, which it sort of is, but it's more a drug that reduces hepatic gluconeogenesis. And we were able to show that it could actually improve the performance of antidepressants in bipolar disorder, maybe also major depression. So we were in this area. We were trying to figure out a way to get insulin into the head. Obviously, intranasally delivered worked. It's kind of a crude way to do it. Wasn't quite so, I guess, so surgical, so to speak. And while we were there at that time, then we started taking an interest in other proteins that could sensitize insulin to be more accessible. And that's when we had the light bulb moment to start looking at GLP-1.
Now, you also taught me something else, and I forgot now whether it was bipolar or Alzheimer's. You called it diabetes type 3. Which was that?
That's right. So that was actually Alzheimer's disease, and we were trying to find a moniker to sort of bring this all together. We know type 1 diabetes. We all know type 2 diabetes. And so some people have referred to Alzheimer's as evidence of type 3 diabetes. 80% of people with Alzheimer's have impaired insulin signaling in the periphery. It's that high.
I did not realize.
It's that high. Now, that doesn't mean that diabetes causes every case of Alzheimer's disease. But we've known forever that ApoE4 allelic variant is a risk. There's another metabolic gene, if you will. And we were also interested in trying to figure out, is there a way we could capture neuropsychiatric disorders, not just Alzheimer's, not just vascular dementia, not just Parkinson's, but adults with major depression, bipolar disorder, schizophrenia. In many cases, part of their impairment in cognition and reward could be explained by this deficit that we see in insulin and related signaling. Now, the history was really interesting, Andy, because insulin came into our life in 1921. That's when it was first discovered as a treatment, as an anti-diabetic. The story around GLP-1 is really fascinating. It actually begins with Ivan Pavlov.
Really?
We all know Ivan Pavlov and his salivating dogs with the bell ringing, classical conditioning, and all of this. We all trained on that in psychology. Well, at the time, he had a view that the pancreatic secretions that the dogs exhibited was entirely mediated by the enteric nervous system. And then what happened in 1902, subsequent to Pavlov's work, two investigators called Starling and Bayliss had really refuted his hypothesis and showed that the stimulation of the pancreas was coming from something locally. And so when there is, in fact, a nutrient stimulus, in addition to having nervous system mediate the pancreas, there was clearly other proteins involved, and they called it secretins. And a person 30 years later called Labar changed it to incretins. And then the first incretin that was really described was GIP in 1970, which is now called glucose insulinotropic polypeptide. Before that, it was called gastric inhibitory polypeptide. And the reason why the name changed is because it doesn't really inhibit gastric acid secretion. It more acts as an incretin. And the definition of an incretin is a protein that encourages insulin release in response to a glucose load. And then, so this is 1970, and then in 1986 is when GLP-1 was discovered. Now, at the time, it was realized that GLP and the GIP act as incretins. They facilitate insulin release. But then it became aware to the scientific community that they inhibit glucagon, they affect the gut motility, they affect satiety, and it wasn't for quite some time later that it was recognized that, guess what? GLP-1 is also produced in your brain. And it's produced in the back of the brain called the nucleus tractus solitarius. It has we were talking about circuits and networks earlier, Andy. It has a circuit that goes up to your hypothalamus, which regulates your eating, but it had a circuit that went to the cognition parts of the brain, the hippocampus, the cortex. Another circuit went to the ventral tegmental area. And we thought, that's kind of cool.
Yeah.
Maybe we should take more of an interest in this. So then we did a postmortem brain study, again, another postmortem brain study, and we found that in major depression and bipolar disorder, these folks who live with these conditions have a decreased expression of the GLP-1 receptor in the hippocampus. Wow. So there's actually receptor alteration in that group. So this was really sort of, for us, was interesting because there's an interesting story about these little lizards called Gila monsters. It's such an interesting story because when you're developing a pharmaceutical and you want the pharmaceutical to mimic a innate or a native or endogenous protein, difficulty number one is how are you going to get this protein into the body? Because our gastric system metabolizes it. So that's solved by giving it parenterally, giving it subcutaneous, and that's how many of the GLP-1s are available. Right. But there's another problem. The other problem was, or challenge I should say, is that in the body breaks down GLP-1 really quickly. In fact, the half-life is only about 60 seconds, 90 seconds. And there's a protein called DPP-4. Right. Many of you have heard of gliptins, like Januvia, drugs like that. They work by inhibiting DPP-4. And by doing so, it allows a longer half-life of the endogenous or the native GLP-1. That's one way you can kind of approach this. The other way you can approach this is just give people a synthetic mimic of GLP-1. Right. But the challenge was, how can we create a protein or a chemical that can mimic GLP-1 that wouldn't be subject to rapid proteolytic degradation? Yep. And so what happened in these Gila monsters, these little lizards, it was discovered in 1994 that in their venom, they have a protein that just looks almost entirely like GLP-1 in the humans, but it doesn't break down so easily. Wow. It resists the proteolytic cleavage, and that was the recipe, so to speak, for the pharmaceutical industry to create GLP-1s, with exenatide being the first one in 2005, and then we discovered that they get into the brain. Yes. And so we thought, this is pretty cool. Because then what happened at the same time, Andy, was that these observational studies started coming out where people were saying these drugs are, yes, causing significant improvement in weight and glucose control, but in addition to all of that, they're slowing the loss of cognitive function in diabetics and maybe even decreasing the incidence and declaration of MCI and Alzheimer's disease. Wow. So we thought, oh, that's kind of cool. So we did a study. Yes. We did a study in some rats. So we took some rats. We did the study in South Korea. It's very hard to do it in North Korea. So we did it in South. we have a group we work with in South Korea. It's much easier to work there. I can imagine. and what we did was we subjected the animals to stress. It was an animal study. Right. And it's called chronic uncontrollable stress. Right. The animal is in a sense of despair. Yep. And then we looked at, in the brain trophic factors. Yep. And we also looked at other elements like brain plasticity, and guess what? Brain trophic factor went down, brain plasticity was altered, and then we pre-treated them with Victoza, one of the GLP-1 drugs, and it completely protected the brain from this insult. Wow. Then we gave the animals steroid to their brain, which is very toxic, dexamethasone, and GLP-1 protected that. Wow. And then we went one step further. We did this little experiment. We determined that it's actually glutamate mediated. So we all know that Auvelity is approved as an antidepressant, August 2022. It's a dextromethorphan NMDA antagonist. It stimulates GLP-1 in the pancreas. Isn't that interesting?
Well, you know, decreased weight was seen in those trials.
Decreased weight is seen. So NMDA antagonists stimulate GLP, and GLP stimulates glutamate. And interestingly, insulin stimulates glutamate, and insulin stimulates GLP. So I always talk about the threesome. Now, when I say a threesome, that means a lot of things to a lot of people. But a threesome to me, means insulin, GLP, and glutamate, and they are interfunctional in how they're playing off each other. So you give an insulin drug, you're giving them GLP modulation, NMDA. NMDA, you're modulating the other two. So this was intriguing to us, and we thought, wow, this must be maybe an opportunity to repurpose a drug.
Absolutely. Absolutely. Well, you and I have talked about this many times. This idea of repurposing, I was reviewing some slides for a talk I'm going to be giving in a couple days with our friend Joe Goldberg.
Yep.
And it's a nomenclature talk, talking about how our drugs are named. And it was a very funny example. Can you imagine if somebody has primary pulmonary hypertension, and I said, I'm going to give you an erectile dysfunction drug. So, because these drugs do many different things.
Right.
And the body doesn't know that you're supposed to be working for insulin and diabetes. The body just says, hey, I got GLP receptors all over. I got to do all this. So this idea of repurposing, I think, is really cool. But a lot of people put things into boxes.
Yeah.
And I remember early in my career, Roger, and you were involved in this too, late 90s, early 2000s, when we started talking about antipsychotics for mood disorders.
Right.
And some people's heads exploded, you know, because they just thought, no, they're supposed to do this. And I remember actually the first time I was consulting with Avenir when they had a form, a dextromethorphan-quinidine combo, Nudexa, and that was for pseudobulbar affect, PBA. And I remember I had done a study with a sigma-1 agonist for depression in the early 2000s, so I knew that. And I saw the mechanism, NMDA, sigma-1. I said, oh my God, this is an antidepressant.
Right.
They looked at me like I was from Mars. No, it's for PBA. So sometimes we get stuck a little bit in our boxes, but Roger, sounds like you're a little bit transdiagnostic or transmorphic in how you think.
You know, it really is. And the way I've been sort of thinking about this is like, I'm an academician, I'm a clinician. I see patients, I have for, 30-odd years. And it has been very frustrating that most of the existing treatments have not been adequate in helping patients cognitively and helping patients in the area of reward function. And we're going to talk about this throughout the next couple of days in some of our presentations, but I think clinicians know that when you talk about what is it about the phenomenology of mental illness that is so debasing of human capital, so decreasing their function, their role function, it really can be distilled down to a couple of dimensions, that being cognition and reward. Well, so big issue.
I would argue cognition and anhedonia are what make us human.
Exactly.
To some degree. I mean, those are almost the essence of humanity to some degree, and that's also what allows us to function, but also to have interactions, social relationships. These are the things that keep us healthy.
You know what's so interesting is that what happened in the GLP-1 area is like anything else. The technology became more sophisticated, the knowledge became more sophisticated, and I think most here know that in addition to having so-called monoagonists like Ozempic-type drugs, we also have dual agonists, which are combining GLP and GIP, the first example being tirzepatide Mounjaro. It's expected next year that we're going to see a triple agonist, that being a GLP, GIP, and glucagon. And a simple, I guess, way of saying it, which may be overly reductive, is that the more that you're agonizing these different targets, the greater the metabolic effect and the greater the effect on weight. In addition to these ways of evolving the thinking with these medicines, what's also happening is that there's different routes. In other words, we now have the oral semaglutide. But next year, there's a drug called orforglipron, which is a non-peptide oral GLP, which you don't need to take, because the oral semaglutide, you can't take it close to your meal and all that kind of thing.
Have some coating or something.
Correct.
Taste.
Correct. So this would not have that issue. It'd just be like popping a pill, it'd be a lot easier. There's also attempts to try and get more synergy with other proteins. Some people may have heard of a combination called CagriSema. CagriSema is a combination of cagrelintide and semaglutide. Cagrelintide is a mimic of what's called amylin, and amylin is also an incretin released by the beta cells of the pancreas. And then one final issue is many might be aware of the fact that when you lose a lot of weight with GLP-1s, you also can lose muscle mass. You can lose lean mass. Now, lean mass is not synonymous with muscle mass. Lean mass is muscle, bones, and viscera, and water. But there are people who do lose mass. Now, losing lean mass is expected if you lose fat mass, because there's less gravitation need to hold the weight. And you would see this in bariatric surgery, you'd see this with any other kind of weight loss approach. Some people started to wonder whether GLP-1s have some direct, somehow deleterious effect on muscle atrophy. That's not quite clear, but what is clear is that many people who take GLP-1s do need to involve themselves in more exercise anyways, but resistance to offset the muscle loss. And there are two proteins, one's called myostatin, another one's called activin. And so some of the newer generation GLP-1s are combining the GLP with an antibody to myostatin, an antibody to activin.
Wow.
These are two proteins that break down the muscle, so it's a way of holding your muscle. And you might be saying, well, this seems like much ado about nothing, but it's not, because elderly folks are at much higher risk of diabetes, and they're more susceptible to losing, sarcopenia is the loss of muscles. That's one issue to think about. But coming back to psychiatry, what was interesting is that we then started to do some what's called proof of concept studies. We took adults who had depression or bipolar disorder. We put them in the MRI scanner, also the MRS scanner. We can look at the neurochemistry of the brain. And we were giving them GLP-1. The first one we tested was liraglutide. And we were able to show after a single dose that we could change the glutamate signal in the reward center of the brain. We measured this with a chemical called N-acetylaspartate. And consequently, we also discovered that when we were doing this, we were seeing immediate subjective reporting of reward improvement and cognition improvement. And then we replicated that more recently. So then what caught my eye then, because this all comes together kind of in a pluralism of evidence, so to speak, where many would know Epic Medical Records, the big digital electronic health record company. 250 million or 300 million people now in their database. They were reporting out that when you prescribe a GLP type drug, you are able to see quite predictably and robustly a lower rate of depression and anxiety in the medical record, and there's different ways they code that. And then to add to the interest, there was also decreased smoking behavior, decreased opioid consumption, as well as decreased alcohol consumption. So I think by now, many would be aware that there are 5 FDA indications to the incretins. Of course, weight and glucose, everybody knows, type 2 diabetes. But to add to that, we have obstructive sleep apnea, we have cardiovascular mortality reduction in obesity, and reducing kidney disease progression in diabetics. Under investigation in phase 3, late phase development, are several others, including peripheral arterial disease, metabolic liver disease, and what's called heart failure with preserved ejection fraction. That's under development. So I just listed 8 conditions that are either on indication or just close to indication that affect psychiatric patients more than other patients. And then to add to the plot, there's a lot of interest in developing these drugs for PCOS. And just to put a fine point, women with bipolar disorder, even before they get valparate, which is a risk, have a much higher rate of polycystic ovarian syndrome. Oral contraceptives and metformins have been used for years. It turns out that the GLP-1s might be better. And in fact, for all of our female patients where we give GLP-1s to, we have to caution them about potential increase in fertility, which could be desired, but in some cases is not. And so on. My patients always say, it doesn't matter, Dr. McIntyre, I'm not having any sexual relations. They always tell me this. I always go through the spiel, and they always say, it doesn't matter, you're wasting your time talking about this. And then they get pregnant. And they say, I don't know how this happened.
Funny thing, you start feeling better, you help your sex drive.
I mean, I know that, and I always tell people, you don't need to go to the church to hear about the Immaculate Conception. Just come to my office. Come to my office. Immaculate Conception happens all the time.
Well, Roger, you actually missed a very big one. I thought you were about to say, when you talked about late phase development of semaglutide, Novo Nordisk, for Alzheimer's disease. Talk about that.
Yeah, absolutely. And this is the part which is really interesting. And that is that, so I'm kind of listing these sort of, going through the chronology very quickly, but also trying to quickly capture some of these, ready for implementation.
It's hard to keep up with all of them.
It's happening so quickly. But I think conceptually there's something really interesting, Andy, and that is that we in psychiatry have a philosophy that we don't do anything until we have a 5-alarm fire. We wait till the whole house is burning down, and then we start to act. And it's obviously a metaphor for most areas of chronic disease. They're in the prevention business. And we are in the treatment business in psychiatry. And why are we not in the prevention business? And what's so interesting about these agents, these GLP-1s, which have very interesting effects in reducing inflammation, oxidative stress, they have effects on insulin and so on and so forth, is they have a preventative effect, a disease-modifying effect. And so I had mentioned that diabetics' cognitive loss is slowed down. And so the folks who make Ozempic, a company called Novo Nordisk in Denmark, they're doing a 3-year study right now in people with MCI or amyloid-positive early Alzheimer's disease, trying to show whether the semaglutide can slow the deterioration. So it's a disease-modifying approach. And so that's conceptually interesting. And it's modifying rather than treating. But it's also obviously of clinical relevance to us in many, many ways. One other aspect, I think, just to finish this off, is that we in psychiatry, there's a couple other targets that we're often asked to treat, like binge eating disorder. And we know that we have amphetamines for that. We have SSRIs like fluoxetine for that. But now these agents are showing nice anti-binge effects. And then just to finish that, I think that for me, we know that many colleagues would commence metformin to treat psychotropic drug-related weight gain. GLP-1s have leapfrogged ahead of that. So to bring this together, I think if there was parsimony here, or just to kind of have a theme to this, what we're seeing with GLP-1s in the human brain is we are seeing a correction of what's called reward salience. So that food chatter that people have who keep eating, the craving for alcohol, nicotine, opioids, what have you, this is significantly regulated when you're on a GLP. It's not dampened, it's regulated. That, along with the fact it has these direct anti-inflammatory other effects, is bestowing all of these benefits, which are so purposeful in our patients. So we are now, we've taken this observation of metabolism and obesity and type 2 diabetes, metabolic syndrome, PCOS, differentially affecting psychiatric patients. We've tried to unravel the pathophysiologic substrate of that, and from that, come up with what is called a target. And we had initially started with insulin. We haven't abandoned insulin at all, but we had a pragmatism in mind, and the GLP-1s lent itself to that. And now I can say that the private sector has now started. We talked about the study with semaglutide and Alzheimer's disease, but now, in fact, there are studies that are going to be taking place in major depressive disorder, in bipolar disorder, schizophrenia, alcohol use disorder, to treat and prevent episodes. And this is not just experimental studies. These are big studies being done.
Oh, these are registration. They're trying to actually get FDA approval for these drugs. And Eli Lilly, actually, I just reviewed the series of protocols. They have 6 studies starting up right now. One of them, which blew my mind, Roger, is cognitive impairment associated with schizophrenia, which Roger and I coincidentally will be talking about tomorrow morning. But it fits in with what you've been saying about Alzheimer's and Parkinson's, neurodegeneration. So we have that. These drugs may not be acute antidepressants, but they're doing studies to prevent relapse for both MDD and bipolar, that's 3, alcohol, opioid, smoking. All starting up at once, all going for FDA approval indications. So I've been saying we must be conversant with GLP-1s, guys, not just because of the antipsychotic-induced weight gain or weight loss. We're really going to be using them for psychiatric indications and for companions. And you know, it struck me, think of how many of our patients abuse substances. So mood disorders in particular are such a disturbance of that salience network, the motivation reward system. And it's interesting you're binge eating. I sometimes think of binge eating disorder as self-medication with food, almost like substance abuse with food. And it's interesting that biologically, these GLP-1 receptors are found exactly where you would want them to help, in the ventral tegment, not just ventral tegment, but in the ventral striatum, the limbic system, nucleus accumbens, prefrontal cortex, hippocampus. The single most reproducible neuroradiologic finding in depression is shrinkage of the hippocampus. And that is one part of the brain where you can have neurogenesis. So it all kind of fits together.
It really fits together, and it's pretty kind of cool, because what we can do now, because we have this incredible molecular technology now, and you can pick a region of the brain in rodent models or other animals, or of course postmortem human brain, And we've done this in human brain where you can look at functional variants of genes and look at receptor expression. And indeed what Andy's saying is true. When you look at the gene that codes for GLP, which is called the pre-pro-glucagon gene, or its receptor, the messenger RNA, which is sort of a kind of a...
Right. Where's it being made?
Yeah, where's it being made? That tells you where it's being made, is really, really expressed in hippocampal structures, parahippocampal structures, executive control network structures, reward structures, like you mentioned. Yeah. And so this is exactly why the prevailing view in obesity research and obesity therapeutics is that these drugs are not just simply causing weight loss because people have, you know, nausea and gut motility. That's not explaining all this. It's much more profound. And it's a CNS effect. And so, but you know, it's interesting because when you look at the history of psychiatry and the pharmaceuticals, there's been a lot of sort of crosstalk with the drugs, so to speak. We poach their drugs, they poach our drugs, so to speak. Like we all know memantine. So memantine is a drug, it's a glutamatergic modulator. We know it better in the world of dementia. But that was a drug, or is a drug, that was synthesized by Eli Lilly in 1968 to treat diabetes.
That's right.
And the reason why they developed it for diabetes is because then and now there is an interest in trying to slow down pancreatic beta cell apoptosis. Because as you know, there's two problems in type 2 diabetes. One problem is that there's peripheral insulin resistance. The second problem is you start to lose beta cell function over time. And so one tactic is to try to preserve beta cell function. Hence the desire to have a therapeutic that can prolong the survival. That's called anti-apoptosis. And that was the thinking, because memantine, by affecting the NMDA receptor, would somehow have anti-apoptotic effects. That was the thinking. It didn't quite pan out with respect to helping glucose. Its role in dementia became a later observation. And there's many examples of this. Topiramate, which has its role in other areas of neurology. We use it sometimes in binge eating or alcohol use. That actually is a sugar that was actually repurposed. It was actually being considered for diabetes.
It was originally developed for diabetes.
That's right.
It was.
And so it got repurposed in psychiatry. There's many examples. And so to Andy's point earlier, and I think that when nomenclature changing, it becomes more and more apparent that the effects that many of our pharmacologic agents have, especially in the signal transduction cascades, are much more overlapping than we'd appreciated. And that's why we're seeing this repurposing opportunity. But it would be pretty cool for us to have an opportunity in psychiatry to have a class of therapeutics that are truly disease modifying. Like with all due respect to the anti-amyloid drugs, which do call themselves disease modifying, I think that's a bit charitable. That's a charitable sort of designation. To say they're disease modifying. To me, disease modifying is not ambiguous. Disease modifying means the illness has stopped. The illness has reversed. That's disease modifying. Slowing it from going 100 miles an hour down to 80 miles an hour, I don't want to be cynical, but that's not to me how I define it. And so that's important, but that's not what we want. We want something that stops it and reverses it. That's disease modification. And so we won't have that type of transformation until we have a better characterization of the pathophysiology. And where the road is leading now is in some of these concepts that Andy mentioned around the neuroplasticity. Some of these molecular systems are all being targeted in a very unique way. What's interesting to us, and it's been kind of befuddling in some ways, is that we just did a study looking at how much of the GLP gets into the brain. You know, the ratio is about 5%. So the concentration of GLP-1 in your head is not very much relative to the periphery. It's about 5% of the concentration of the periphery. So it is getting in. They're big molecules. They can't quite get across the blood-brain barrier as easily. But they're getting in. And so that's an interesting sort of more of a kind of an engineering issue. But that seems to be the case, although it's still being debated whether that's anyway, if that's entirely the case.
There are ways to address that. There are some drugs in development right now that are, there's various kinds of formulations that can improve crossing the blood-brain barriers, you know. I want to say something else. You talked about this idea, how much of their effects are peripheral, how much are central. And so I think it's very clear to me, well, certainly losing weight, which you taught me this years ago too. Weight loss by any means is an antidepressant. I remember you teaching me that in inflammation. But clearly that's a good thing, but clearly there's also something centrally going on. So it's not either or, it's sort of all of the above.
This is such a critical point because, as I mentioned, you all know the DPP-4 inhibitors, like these are the gliptin-type drugs. And when people have looked at the GLP-1s and the DPP-4s, well, again, the GLP-1s are mimicking the endogenous GLP. The DPP-4s are inhibiting the enzyme that breaks down the native GLP-1. When you look at the brain therapeutic effects of the incretin classes of drugs, we see nothing but a compelling story with the GLPs, but we're not seeing a compelling story with the DPP-4s. So that tells us that it seems like it's not just peripheral.
No.
It's got to be something central.
I see what you're saying.
And also there clearly is not enough of the native or the endogenous GLP-1 to do the trick, so to speak, because by boosting the native or endogenous GLP-1 with DPP-4, it's not having the same brain therapeutic effect. It's having some, not quite the same effect. So the synthetic GLPs that are getting into the head, they seem to be much, much more robust. And again, as you add more targets, like you add GIP to it, it gets even more interesting because GIP is also in the brain. So it's very interesting.
I forgot to do something here, Roger. This is a CME talk, so guys in the back, Marlene, we do have to advance the slide one. We have to show our disclosure slide, so I just want to make sure we all get our CME here. And we're going to have time for questions. Hope you've been typing in your questions into Slido, but I'm going to take the chair's prerogative to ask Roger the first question. Actually, I have two. The first one is, clearly, it sounds like these drugs are neuroprotective, but aren't they also pro-cognitive to some degree? I mean, can you see improvement in cognition, or is this more of a prevention of the degradation?
It's a great question. Right now, the evidence certainly suggests it's both, that you do see a reduction in the progressive cognitive loss. There now is evidence in rodent models, and now we have some data in our lab to show that there's a direct pro-cognitive effect. Now, it's a little too early to sort of really have a summary or even a final statement on this. But if I was to actually just make an interim kind of report, we're always making interim reports, where I think this may not surprise anybody, where in fact the signal looks much more robust, reproducible across the different agents is helping reward function, like anhedonia. Is it helping cognition? It absolutely is, but we're seeing more signal. This is interim. And, things can change. Things can change. And that doesn't surprise me in some ways, right? And so there's different facets to reward, the value you put on a reward, the motivation, the valence, the anticipation, the learning around reward. All of this is encompassed by what we call hedonic tone, and that seems to be, at least in the work we've done, others have done, the animal work, that seems to be more robust. But we are seeing a pro-cognitive effect. There's no question about that. Now, and related to this, I should be very clear, is that, whenever we sign up to give someone a medicine, the first rule of the day is do not harm the patient. And so when we look at this, one of the questions that came up about 2 years ago was, do these drugs increase the risk of suicide?
Yes.
And some might say, well, hang on, doesn't that kind of rain on the parade? How can that all, how does that all come together? Well, whenever you are targeting reward systems, there's idiosyncrasies that can happen, and it's possible that in some idiosyncratic situations, you could actually have a worsening of mood or mental health. So to summarize, the last 2 years, there's been over a dozen what's called observational cohort studies, over a dozen pharmacovigilance studies. The FDA's looked at this, the European FDA called the EMA's looked at this, the UK FDA's looked at this. In other words, it's been interrogated, and we've done a lot of work on this in my center as well, and we cannot find evidence of causation. It's an association, but not causation. But I think for now, Andy, I'm still sort of the view that it's not off my radar screen yet.
Yep.
And we know from our own experience with bariatric surgery, some patients can have very untoward psychiatric outcomes. Not common, but it can happen.
Sure.
And for those maybe a little less familiar, there was a drug that was approved in Europe maybe 15, 20 years ago called rimonabant.
I studied that drug
Rimonabant.
for schizophrenia. I'll tell you
Interesting.
that
That's an
later.
inverse, as you know, inverse partial agonist of the cannabinoid 1 receptor.
That's right.
And it was taken off the market after 2 years in Europe. It was a weight loss drug because of amplification of depression and suicidality. So we've been to this road before, but just as an FYI. And by the way, we're going to have a very detailed discussion about these aspects of GLP-1s and others. I think it's our Saturday afternoon presentation. So we're just kind of giving an overview right now, but we're going to get into the details of this.
Great. Well, we have a bunch of questions submitted. I want to thank you. So here we go. Roger, what do you see as the future of GLP-1 research? Well, that's a big question, but let's narrow it down to what excites you the most.
I think what excites me the most of the GLP-1 science, the GLP-1 pharmacology, the results from epidemiologic studies, observational work, is the possibility of disease modification, that we could prevent the onset of mental illness like depression in people who have diabetes, obesity. But for people who do have already declared the illness, major depression, bipolar disorder, is it possible that we could be offering them some degree of illness resiliency enhancement such that the relapse risk is so much lower? Andy made reference to studies that are now going to start in depression, bipolar, and schizophrenia. Big phase 3 studies, folks. These are for registration purposes. These studies are not trying to determine whether GLP-1s treat mania or treat psychosis or treat depression. Instead, they're trying to prevent the relapse from happening. Because if you look at all the evidence in its totality, it seems as though the benefit is modifying the course. And that would be pretty cool. That would be pretty cool if we could do that. As I said, really, truly disease modify would be pretty interesting. To me, that's the most exciting.
That is super exciting. And, something else we didn't talk about is these drugs don't all work the same. I mean, simply calling a drug an agonist is one thing, but, I did research training on dopamine receptor pharmacology 30 years ago. It was pretty straightforward then, but my God, now we've learned so much about once you bind, there's so many different things you can do, and it turns out the two commonly used on the market, semaglutide, tirzepatide, have differences in how they act at the GLP-1 receptor. Maybe you could talk a little what does that mean?
It's so true, and I think that you're right, Andy. Back, in the day, when we talked about a drug binding to a receptor, it either blocked it, it stimulated it, or it partially stimulated. That was all we needed to know. Now what we've learned is that when a drug binds to a receptor, for example, a G protein coupled receptor, when it binds to a receptor, it ignites a host of cascades underneath the surface. So many cascades are activated. And some very smart people realized that you can then develop drugs that have bias activations. They activate this cascade, but not that cascade. And some might say, well, this seems like much ado about nothing. What's the takeaway from this? Well, it's huge, because if you activate this cascade, that might maybe reduce the risk of a safety issue. If you activate this cascade, you might get the good without the baggage, so to speak. And this has now been done. So tirzepatide, Mounjaro, is different than Ozempic in, two ways, one of which is a dual versus a mono. But specifically at the GLP-1, it's quite different. You see, semaglutide just activates the receptor, and so many cascades are activated. Tirzepatide's biased. It biases. It does not activate what's called the beta-arrestin pathway, and that's thought to result in greater expression of the GLP-1, which is thought to be why it has greater weight loss. Now, this has found itself. We're going to talk about xanomeline tomorrow, which also has biased agonism at M1, M4. We're going to talk more about that tomorrow. And I have a presentation on psychedelics at this presentation. I'm going to be wearing my psychedelic jacket during that presentation. And there's an interest in developing what's called biased agonists of the 5-HT2A receptor. These are non-hallucinogenic hallucinogens. So this isn't just some, pet project of a very keen medicinal chemist. This is prime time in terms of developing drugs this way.
This is a very active area of research. Stay tuned, guys. So it turns out I went to an amazing lecture at ACNP a guy who was an authority on serotonin receptors. He told me there's at least 7 different intracellular signaling pathways. And now with some of the technology, the precision, you can now bias down 1, 2, 3, 4, 5. You can play around with exactly what you do inside. So stay tuned. All right, we got another question here. How do you counsel patients to maximize gut health? We didn't get into your whole interest in the gut biome.
Didn't get into that too much, and this is interesting itself, and you've all heard, of course, about the interest in gut biome and biota and the diversity, and that's a very interesting story. It's already moved into clinical implementation with Clostridium difficile and fecal transplantation. I mean, that went from being kind of a cultish experiment to something that's really a standard of care. And these studies are now going on in people who live with mental illness. People are doing these types of studies. We did some work on this ourselves. Absolutely. I think it's part of a larger jigsaw puzzle. I don't think it's the only piece of the puzzle. Is it a big piece? Is it a small piece? It's still to be determined. But I don't think it's a non-issue. And it's known that GLP-1s and many of our drugs do affect the gut biota biome. So I've always thought this is especially interesting, the gut. And for now, I think we're still looking for a well-balanced diet. People are now looking at ketogenic diets, things like that. That's all interesting. But look, I think the devil here is economics. Our patients can't afford a good diet, and they can't afford GLP-1s. So that's an obvious economic determinant. But I think it's a really, really good point.
Something else you pointed out to me once, exercise actually improves the gut microbiome. So there are straightforward things you can do.
It's truly fascinating. You know, it's interesting. I often tell this story. I mean, some of the stories around medicine are so interesting. John Harvey Kellogg, of course, famous for his cornflakes, had his institute up there in Michigan, in Battle Creek, Michigan. And he was one of the first ones, actually, to have the prescience, the foresight, at the time would have been thought pretty crazy, but he was actually getting the patients to eat yogurt. But then he realized if they actually used the yogurt in the rectum, it was more anxiolytic. Now, that's a complex set of, there's some confounding factors there. But that being said, in some ways he was ahead of the time. Now we're in fact looking at manipulating the gut biome, but he was already doing that quite some time ago.
All right, very good question here. If GLP-1 agonism reduces reward salience, wouldn't that have a negative effect in terms of anhedonia, causing more anhedonia? You talked a little about modulation there. Maybe that has something to do with it.
It's such a great point. And obviously we don't want people to have anhedonia. Interpersonal anhedonia, sexual anhedonia, food anhedonia. Who wants anhedonia when you're watching Emily in Paris? Nobody does, okay? We don't want anhedonia for anything. But at the same time, you don't want to have maladaptive, overreactive reward system. GLP-1s do not dampen the reward system. What they are doing is they're regulating it. So rather than having that overactive rumination, craving, food chatter, where you have an anticipatory reward far exceeding the consummatory reinforcement, that's what propagates the maladaptive behavior. You're instead having a more corrective reward anticipation, which is matching the experience. So the prediction is what you're getting, and that leads to more adaptive eating and adaptive consumption. So it's not taking away, it's actually resetting the reward system.
Well, I'll tell you my personal story. I have type 2 diabetes. It runs in my family. Thank you, Mom. And I am on Ozempic, semaglutide, and I can tell you from personal experience, it shuts off the food noise. That's a great way of saying it. And I can still enjoy food very much. So it has not completely removed the ability to enjoy things. Okay, finally, are the pro-cognitive effects only seen in people with obesity?
No, and what a great question. That question is conceptually and clinically so interesting, and the answer is no. Now, if I was to say this, people who have obesity, are they more likely, or are they, when you look at the effect size, it's greater, but it's not confined to that group. And that's so important. And that's, again, a critical point. Andy, you talked about this. Is the benefit on the brain a byproduct of the peripheral loss of weight?
Well, that doesn't hurt.
Sure. And or is it a direct effect? And the answer is it's both. It's a direct and indirect. The answer is yes. But you don't need to have a BMI of 25 or 30 to have this beneficial effect on cognition and reward.
And you've showed, of course, too, that obesity is associated with cognitive impairment. This abdominal fat, of course, is very pro-inflammatory, releases a lot of cytokines that get across the blood-brain barrier. So obviously losing some of that would be a good thing.
Absolutely would be a good thing. And one final comment, we're going to wrap up in a second. I'm fully aware of the fact that, you know, these agents are very pricey. Access is an issue. We did a study last year, we published it. We did find that people who are using online pharmacies or compound pharmacies, they are much more susceptible to accidental overdose because of self-administration. So there are these aspects that we're fully aware of, and so that is not, that information is not lost on us. We're aware of that, but hopefully those gaps will be sorted out.
Well, unfortunately, that's all the time we have. That was our last question, but Roger, what a journey it's, oh, I said the word. What a story. What an experience you had. Oh, I can't wait to say that. But anyway, Roger, thank you for sharing that.
Thanks, Andy.
Guys, a lot more to come.
Thanks, folks. Thanks for joining us, and we'll look forward to seeing you tomorrow.
Thank you so much, gentlemen. Thank you. And thank you guys so much for getting here a little bit early so you could enjoy dinner with us tonight. Let me give you a couple of quick housekeeping notes. So you will get an email evaluation from NEI each day. So you'll get one sometime later tonight, and it's just to tell us what you thought about each program and each day. So at the end of each day, you'll get a full evaluation on everything you see tomorrow. So just keep an eye out for those in your email. If you're not getting them, stop by the help desk and we'll help you figure that out. You can also find them just by going to the QR codes, and a couple of places in your program guide have them listed. You can proactively go and find those evaluations. Having said that, the way you get your CME is at the end of all of Spring Congress, which is Saturday after lunch. You will get the final evaluation that has everything that you need to complete to get to your CME certificates. So that's the big one to keep an eye out for. And again, we're providing the QR codes. If you're not getting the emails, we want you to know that it's going to be an easy process to find your way there yourselves. So that'll be at the end of all of Congress when you get all of your CME, including the CME for tonight. End of day reminders, we reset this room, we will reset the CME rooms each night, so please don't save seats and leave anything behind. Take everything with you. If you have time right now and want to participate, here's something a little fun. We have a faculty feud coming up in a couple of days.